Welcome. Good. Well, thank you for coming and joining us on such a beautiful afternoon. I always feel incredibly grateful when people decide to leave this beautiful natural environment that we have and come and listen to a science lecture. But tonight you are in for a treat because we have Dr. James Godwin with us, who, as many of you may know, is studying regeneration and actually scarring in this wonderful creature called the axolotl. So we're very excited to share a little bit with you about what that's all about. But I wanted to just, quickly talk a little bit about scarring and kind of ask you all, what do you think of when you think of scars? Like, what is the typical thing that you think about? Skin your knee, right? Right? Cut yourself in the kitchen. That's what I always do. Surgery, skin cancer. Yeah. I mean, we typically think of it as being kind of a cosmetic thing, maybe, to our skin, right, on the outside. What we don't often think about is what happens in the context of disease on the inside, right? So what happens when you have a heart attack, for example, right? That tissue is deprived of oxygen. And then what happens to that tissue? Exactly. A scar, essentially, right? That's sort of forming on your organs. And we often don't really think about the impact of scarring on our organs and on our internal tissues. And so it becomes a very serious problem when we really think about how it impacts health in that context. So tonight, James is going to lead us through just what I think is a really exciting story. James is a trained immunologist, so his background, he's very interested in the role of the immune system in regeneration. I think all of you know that as humans, we don't necessarily regenerate all that well. We do a little bit, but not to the extent that some of these amazing organisms that we work with are able to do. And so he's very interested in comparing and contrasting what we see in some of these animals in nature with what we as humans, the limited ability that we as humans have. And so he has a very interesting role. He has a joint position. He's here at the MDI Biolab as an assistant professor, but he's also at the Jackson Laboratory, and he's doing some of that comparative work in mammals, working with mice there at Jax. And so I'm going to just turn things over to James. You will notice that he does not have a Maine accent. It's really shocking, but he did a lot of his work. He was at the Australian Regenerative Medicine Institute. I don't know if I'm going to get that quite right, at Monash University. But then we persuaded him to come to MDI. So James, thank you so much for being with us this afternoon, and we are excited to hear your story.
Thank you, Jerry. So I may not have an American or Maine accent yet, but my daughter does. But I'm trying to make her watch Bluey so that she can kind of get a bit of an Australian accent on from that. Well, so as Jerry mentioned, I study tissue repair and regeneration in two species. And at one end of the extreme, we have this salamander, which you're going to learn about a bit today, that can do these wonderful, amazing things and repair almost any tissue scar-free. At the other end, you have, did you guys hear me okay? Are you? We good? Okay. All right. There we go. We good? Okay, so as I was mentioning, like at the other end of the extreme, I like
Still not thinking.
Yeah.
now, normally they can control the volume in their whole room. Put it up.
Try swallowing it.
Yeah. Okay, is that better? All right, good. So as I mentioned, we have a wonderful axolotl at one end of the equation that can do all of these clinically relevant structures that I'll talk about later today. And at the other extreme, we have us, pathetic, poor regenerators prone to scarring. And so that divide between these super regenerators and these poor regenerators is really where I spend most of my time trying to fill. That's a bit too much now. Okay, and so what as Jerry mentioned, like scarring seems to most people kind of like a cosmetic thing, right? But actually it's way more serious than that. like scarring accounts for about 42% of all deaths worldwide. And so if scarring happens on the inside of your body in sensitive organs or tissues, it can really be disastrous, right? And so like part of that example would be if you had a cardiac arrest, temporary loss of blood supply to the heart muscle wall, that gets damaged, and the necrotic process eliminates that muscle, and it gets replaced by this ropey, thick, non-stretchy scar tissue. And you may not die straight away, but the survival rate 5 years later is pretty low. And so that takes out a lot of people. And so if it happens in the brain, like a stroke, that could leave you permanently disfigured or paralyzed. If it happens in the spinal cord, that'll form a physical and chemical barrier which blocks the nerves from reconnecting. So you lose the use of your legs and you end up in a wheelchair at best. And so there are other things that can go wrong as well. So if you have, say, a chronic insult that causes damage to your kidneys or your lungs or other organs like the liver, that scarring can impair the function, putting you on the organ transplant list and really compromising your health. And of course, things like childbirth and cesarean sections can even go deeper into causing infertility when complications take place. And of course, although scarring of the skin, can be somewhat cosmetic, of course, if that happens in a child with pouring a kettle on their head or pediatric burns, the emotional trauma associated with that is horrible. So it really does span the full spectrum of death to disfigurement. And so trying to find a solution to this has been really difficult. And there aren't any treatments available on the market. People have been looking for 20, 30 years, and it still hasn't happened. And so today I'm going to talk to you a little bit about how the salamander may be able to help us, guide us on that path. And the hero of the story, second hero, not just the axolotl, is this guy. This is my favorite cell type. And it's called the macrophage. And in Greek, it means big eater, right? Just like my little daughter. And the thing is that this cell is really quite famous for being the front line of defense. The Nobel Prize was awarded back in the 70s for the discovery of this cell type. And the thing is that it runs to the site of injury or infection, and it either eats the bacteria or the fungal cells or the virally infected cells. And it does all this cleanup and it does all this maintenance. It's involved with repair. We've known that for a very long time. But we're starting to understand that it has more functions outside of just pathogen control. It has all of these functions in repairing tissues and really directing the show. And what I'll talk to you about today is that it goes far beyond just inflammation and setting out all the redness and all of the swelling that takes place and cleaning up the mess. They're actually actively involved in building the tissues that form us, right? And so over the last few years, what we've discovered is that these macrophages are like right where all of these organs develop. So they're concentrated in the developing brain, in the developing kidney, in the developing lung and heart. And so what's really interesting is if you get rid of macrophages in early stages of development, you can get like shrunken organs, like really tiny kidneys. And if you give the proteins that are going to expand and make macrophages grow like crazy, you actually get bigger organs. And so they're actually involved in building our body plan, which is pretty interesting because I've been studying them from the angle of regeneration, thinking that, not so important in development, but they are. And so when we try and understand what happens to our bodies, I mean, if you think about our bodies, they're there, and then what happens is throughout our life, we have injuries and we're trying to maintain our body plan and our tissues. The macrophages are there. They're right in various organs, helping maintain it. We call that homeostasis, right? Keeping the status quo, if you like, of the tissues. And so what's fascinating about this is that as we age, our tissues fall apart, right? I mean, as you get older, things don't work very well. And we don't know what the root cause of aging is, right? Part of what MDIBL does is try and solve this problem of aging as well. And aging and regeneration are all kind of interconnected because you regenerate really well when you're young and you kind of lose that ability when you're old. But is that directly responsible? Are they directly connected? Well, macrophages might be that bridge, actually, because what we find, and we published a paper in 2011, I think it was, about cardiac macrophages. With aged mice, the types of macrophages you find in the heart changes with age. And so this is an example of what happens in mice and humans, is that, you know, when organs develop, we have these very embryonic pro-regeneration, pro-building macrophages with a primary focus of building the organ, and they don't really care about pathogens, right? Because there's no pathogens in the embryo in most cases. But what happens after birth in mice and just before birth in humans is that the macrophages start to be born from an alternative location. So instead of coming from the liver, they start coming from the bone marrow, and that bone marrow really takes over the show, right? Once you're born, the bone marrow is the source of all these monocytes, and they enter the blood and they go into the tissues and they replace the old ones with the new ones. But their focus isn't repair. Their focus is pathogen control. And so we kind of have a problem because you need both, right? You need both. And it seems like in mammals, there's some sort of specialization. But interestingly, in animals that regenerate, like the zebrafish and the axolotl, as adults, all of their macrophages seem to come from the liver, right when humans and mice can actually repair pretty well. And what happens in later life is that the bone marrow macrophages kind of dominate. And that's associated with all this scarring and you know, pathogen clearance. So somehow the axolotls worked out the trick because they live in, you know, fairly, you know, pathogen-rich water sometimes, right? And if you chop off their arm, put them in the water, they're fine. So they've got really good pathogen control when it comes to bacteria and yeast and fungus and various other things, but they can maintain this ability to repair. So that's really fascinating to us, and we're trying to work out how they can have their cake and eat it too. And so when we look at the axolotl during development, what we find is that, and this is a 3D illustration of an axolotl, and this is a real 3D image of an axolotl that's been tissue cleared and then using the endogenous transgenic reporter with red macrophages, green oligodendrocyte kind of cells that wrap around nerves. And then in yellow, you can see the axons of the nerves that go from the brain all the way down the spinal cord. This gives you an idea that, well, I'll go back one, sorry. This gives you an idea that the macrophages are kind of close to a lot of these developing tissues. And if we amputate the tail of one of these animals, what we find is the red macrophages swarm to the tail. And over here is the real image. This is a live image where we can actually see this process taking place in front of our eyes. And this is a picture of a live axolotl embryo. So we can track these macrophages, where they're born, where they're located, and then watch them move into the injury site and understand how they're involved in repair. And so we can take that further, and we can use much more sophisticated microscopes that can look at this at high resolution in 3D using a light sheet microscope. And in this video that I'm about to show you, it's a developing embryo, and the macrophages are born in the liver, and you can see they're really sort of enriched in that location. And this is the limb bud, and you can see that they're there before any skeletal structures take place or form. And if you look, here's the heart, you can see they're right where the developing coronary vasculature is forming. And all of the developing nervous system has these kinds of macrophages sort of dotted along their extensions. And what's really interesting is that we just identified this week a brand new stem cell compartment, potentially, where we see macrophages in the caudal hematopoietic tissue, which has never been described in the axolotl, but is present and dominant at early stages of zebrafish development. And so that forms a bridge between our axolotl and our zebrafish. And we can start to take that further. We can use these types of 3D imaging modalities to look for associations between progenitor populations like stem cells and our macrophages during development or during regeneration. And so it's a really interesting system. And of course, you can study, macrophage development in lots of different animals. Probably not with this level of sophistication in some ways. But the reason why we want to do it in the axolotl is because these are the champions of regeneration. These animals are remarkable. They have the ability to regenerate a range of clinically relevant structures throughout their lifespan, like as full adults, after they've had several spawnings of offspring. They can still replace parts of the liver. You can cut off large sections of the heart and they'll survive it and it'll repair. You can sever the spinal cord or take out sections of the spinal cord and they'll repair that spinal cord without scar tissue. And so that's a process we're getting more and more interested in. They can also recover fully from injuries to the brain, like large regions of the brain can be damaged and they can repair that, along with parts of their retina and their other ocular tissues. There's a paper out there showing they can regenerate parts of their jaw when that's cut off. It's not the part I would like to cut off, but their skin and of course muscle and tails, but most impressively, they can do whole limbs. And so you can cut it off at the wrist and they'll regrow a hand. You can cut it off at the upper arm and it will regrow the full arm. So it's very precise and it's very faithful to the original. Sometimes they come back with the same spots and bumps. And how do they know? Like, how do they know which bits regenerate? And how do they mobilize these cells? And why don't humans? This is crazy, right? So this is what we study. We're really trying to get to the heart of what makes them so special. And of course, there's lots of different things to study in these animals. There's lots of things that we can do in terms of comparative biology to find what makes them remarkable and different from mammals. But one of the focuses that I have has been macrophages. And as I mentioned, the reason why they're so important, and as an immunologist, like the immune system's always the center of the universe. And it makes sense to me because they're the cells that are first arriving on the scene. And so they're directing the early events. And so whatever happens after the early events obviously could be dependent upon them. And so back in 2013, we identified that when you get rid of the macrophages and do a limb amputation, you actually, it basically comes back perfect pretty much every time. But if you do that in the absence of macrophages, you get this like thick, ropey scar tissue just below that skin. And it's completely human-like in its repair. Interestingly though, if you then wait till that macrophages come back, because macrophages are born on a regular basis from hematopoietic tissue, and then you re-amputate, the arm comes back perfect, right? So as long as you cut off the scar tissue and the macrophages are back, you're all good. And so we did a similar thing in the heart because, is this a limb-specific problem or is this something a bit more ubiquitous? And so we did that in the heart. And what we found is that in this animal where you can cut off a third of the heart and have it repair, if you get rid of the macrophages, you end up with this horrible scar tissue that's there for we checked almost a year later and it's still there. And the animals are struggling, but it's there. And so it was kind of disappointing that it never fixed itself, but it showed that it's kind of, you have a question? Yeah.
If you reintroduce the macrophages out of scar tissue, are they able to regrow and live?
Yes. So we did that experiment in our PNAS paper. You wait 14 days or 20 days for the macrophages to repopulate the animal, reamputate, and regeneration takes place as normal.
How do you suppress the macrophages?
Good question. So these experiments were done through a mechanism known as quadrinate liposomes. And so what you do is you basically take this caged poison and put it inside a liposome. And basically the macrophages are phagocytic. And so when you inject it IV or IP, the macrophages selectively take it up. Some neutrophils can take it up on rare occasions, But the secret is that it's macrophage specific because they have a low pH garbage bin. And it goes into the low pH garbage bin, which activates the caged poison, and then they all die.
Kill them.
We kill them, yeah.
Situ.
We kill them in situ, that's right. And so you'll hear a little bit in a moment about a genetic system that we built that allows us to use a small molecule to basically, rather than kill all macrophages, we can kill subsets of macrophages. And so we built that here, and Gabriella Johnson in my lab, a Ph.D. student. And through that, we actually got some really cool data in the tail, which I'm about to show you in a minute. And so, but before I do that, I just want to keep going with the scarring angle for a moment, right? And so what we find in both cases, the heart and the limb, is that in the absence of macrophages, you get this scar tissue induction like humans. So that doesn't make a huge amount of sense because we have macrophages and salamanders have macrophages. How come theirs are like blocking this process? In fact, in mammals, it seems like macrophages add fuel to the fire. They're actually, in many cases, part of the pathway that activates scarring. And so fibroblasts are these kind of cells, connective tissue cells that are spread throughout multiple organs throughout the body. And they're kind of like the glue between lots of other cell types. And unfortunately, in a wound scenario, they get corrupted in many cases, and they start to become scar-producing cell types that form the scar tissue. And a lot of this is through this mechanism with this protein called TGF-beta. You don't have to remember that, but when we add TGF-beta to these cells, they spontaneously turn into these bad guys, which are shown here in green. And when we add macrophages just by themselves, mouse macrophages on mouse fibroblasts, without TGF-beta, they're making the TGF-beta and they're actually causing the scar tissue. And when you add them together, it's just twice as bad. But surprisingly, in the axolotl, the fibroblasts can turn into bad guys, and they do, obviously, in the absence of macrophages. And when you add macrophages by themselves, they don't turn into myofibroblasts, and most importantly, they can attenuate the process of TGF-beta-induced myofibroblast induction. So that implies that both mouse and salamander macrophages produce the poison, but only the axolotl produces the antidote at the same time. And so we're really trying to identify what it is that can give the axolotl macrophages this power, the superpower of being able to suppress this fibrotic activation. And so once we find that, there is a clear path for the clinic. And the path would be already established. In fact, there's about 9 clinical trials that have taken mouse or human macrophages and modified them with a cargo, a therapeutic cargo. No one's attempted it for scarring. And if we were able to put the right cargo inside the human engineered macrophages, we could deliver them to humans to attenuate some of those profibrotic activity during injury. And so I mentioned that macrophages are important in developing organs and that they're associated with many of the developing systems in the axolotl and the mouse. And so we were interested to know if they would control spinal cord regeneration. And the axolotl, as I said, is really powerful in terms of its ability to regenerate spinal cord. There's a few ways you can do that particular experiment. One of the methods that we chose to do is to basically do a resection of the tail, similar to what I showed you in a previous slide. But we actually make sure that we cut through the spinal cord. And so normally after amputation of the axolotl tail, you get this beautiful regrowth associated with like myelination of the peripheral nervous system. It's kind of like, think of myelination as like the insulation that you put on copper wires to make the copper wires work better, right? So they don't short circuit. We see this ependymal tube extension, and underneath that, shown here on this slide in blue, is brand new skeletal tissue, right? And so we see that every time, and we have the ability to measure that. And we also see in green here this beautiful extension of the central nervous system, the spinal cord, inside this ependymal tube. And what happens when we get rid of macrophages genetically using our CD68 ablation tools, we find that all of those processes fail. We can't find any evidence of scarring because there's not many fibroblasts in that area. It's pretty tight with skin and epithelial layers and spinal cord. And so there's no real scarring that's the problem here. And that sort of led us behind the curtain, because what we've now revealed is that macrophages are involved not only in fibrotic control, but also the differentiation and development of stem cell populations that give rise to those new tissues. And so the myelination process, you can see all these pink dots here, rather than being organized in beautiful and associated with neurons, they're just disorganized. We see a lack of this tube extension. So there's nowhere for the spinal cord to grow into. We see a lack of the spinal cord there. We see these sort of axons in general just stunted. And so when we quantify this, it's remarkable. It's just literally a full stop. Nothing grows. And so that gives us the impression that macrophages either control all of these processes independently or potentially as a collective in some sort of interactive domino effect. And so that's our next question that we have to address. And if we can identify what it is that allows macrophages to direct traffic and tell each stem cell population which structure they're going to give rise to, that could be immensely valuable for regenerative therapies. And so if we can find that information, we can then put that inside our Trojan horse macrophages, right? And so if we know the interactions that macrophages have with different neural populations, vascular populations, or even skeletal populations, we could arm them with the right instructions and either inject them directly or put them into the circulation and use their natural abilities to home to the site of injury, because they're attracted to injuries, right? They sniff it out and they literally can travel from one end of the body to the other, hunting down the injury, right? And so that really allows a few different modalities. I mean, of course, it could be expensive, personalized medicine initially, right, to do the proof of concept, but eventually you could make IPS generic off-the-shelf versions that carry a suicide gene so that you have them ready in a hospital environment, deliver them to a patient, give the patient a drug. The same drug that we use for our ablation technology could then kill them. Because that drug's non-toxic and it's possible to use that in humans. And they've done that with human T cells. And so that really wraps it up with some of the data that I wanted to talk to you about. But I want to talk to you about the team that helps me do this work. In particular, Gabriella, Gabby, as we call her, she's a wonderful PhD student who has generated all of the macrophage ablation technology in my lab and deployed that to the spinal cord. And she even generated that beautiful 3D image that I presented today with help from Frederick, who edited that and battled the software and the processing time that it required. That image took about 2 weeks to transfer across our internet connection from Bruker. But yeah, and the rest of my team that made this possible. But most importantly, none of this work would happen without our donors and our institutional support and our federal support. And so thank you to all of you who contribute to this work and keep our wonderful cause supporting this work. And so that's it. Last thing I wanted to put up here, you don't have to watch the whole thing, it's really long, so we'll talk over it in a minute. But a lot of people ask me, like, why is it that you think salamanders can regenerate their arms? And I'm like, well, there's lots of, it seems strange. But this is a picture of an axolotl. She's laying eggs one by one, like tinsel on a Christmas tree. And they're all really well spaced and aerated. And she's using her legs to position them. And it takes her a while. She's really dedicated. Not only is she dedicated to spacing out the eggs, like for the survival of her species, but yeah, that could be one of the reasons why they need that. I mean, there's also another part of the mating ritual uses the arms, but I won't go into that. So, I guess that why do they regenerate the heart? I mean, that doesn't make sense, but there could be some common modules that link all the different tissues that regenerate. And maybe it's just they've got cool macrophages. So, yeah. So it goes on like that forever. She literally takes like hours, and then we collect those and we inject them with DNA and make transgenic animals and stuff. So that's how we do it. Yeah, and so that's it. So I'm here to
I've got a bunch, so it's for you.
Good. I remember some earlier work, which older people like my other brother Daryl probably will remember better than I do. But that there was a real interesting dependence. I think this was in terrestrial salamanders between the nerves and the muscles. That a denervated limb would not regenerate or something.
correct.
Do you see that same sort of interdependence?
Oh,
with
yeah.
the axolotl?
Yeah, so that's the thing that's really interesting. I obviously don't have time to present that today, but so it's been known for like, I don't know, probably 70 years now, I think, that the nerve is kind of essential for a lot of different regenerative contexts. It was kind of a bit mysterious. And wasn't really known what it is that the nerve supplies that makes it compulsory for regeneration. When I was working in London in Jeremy Brox's lab, we sort of stumbled across a molecule that was, we were studying another process, and we discovered this molecule that was expressed around the nerve at the time of injury. And we tested that, and it turned out that we could actually substitute for the role of nerve by cutting the 3 nerves that feed into the arm, and then putting that protein or that gene in there. And we got floppy arms because they didn't have any, innervated muscle, but they regenerated the pattern. And so we've been narrowing in on that signal for quite some time. At the time, we thought it was the green cells that wrap around the nerve, the Schwann cells, right? And it turns out that, like, as luck would have it, it's not coming from the Schwann cells. Now we have more sophisticated technology than back then. We have single cell RNA-seq, where we can tell who's making the protein, and all of our immunostaining was a false positive because the macrophages that come in are eating the Schwann cells. And so when you stain them with an antibody, we thought they were Schwann cells, but actually they're Schwann cells they're dead Schwann cells inside the body of a macrophage. And so that signal was coming from macrophages, right? And so I made a transgenic mouse, that overexpressed that molecule, and it actually increases regeneration and the types of things that can regenerate. So yeah, it's an important axis. Macrophage and nerve interactions are really important. Yeah.
When I was in medical school, half a century ago, macrophages were classified in M1 and M2 macrophages. They have different characteristics, they behave different.
Sure.
And one of them was involved in the phagocytosis. And I can't remember which one it was.
Right.
But the other one, nobody knew what it was doing. Is the one that we didn't know what it was doing the one that you're addressing here?
Well, depends on my answer would depend on the audience, right? If I was at a macrophage conference or an immunology conference, and I even mentioned M1, M2, I'd probably get yelled at by 50 people. But it's a useful paradigm to kind of represent extreme states of what macrophages can do. And the idea is that, they come in neutral, they're exposed to tissue damage, they get hot, they get inflammatory. And then at a certain point, they make a decision to sort of calm down the inflammatory pathways and then differentiate into different subtypes. And it could be like M2A, B, C, D, E now, at least, right? There's all sorts of different situations. So it's more of a, and it's linked with metabolism and function, depending on which tissues. It's fairly complicated. So there's lots of different subtypes, but like for instance, one of those populations may turn into a bone-eating cell, like an osteoclast, if given the right priming signal and then a secondary signal. And so the whole M1, M2 thing is really fascinating from a scarring perspective, right? Because the M2, even now, M2 is thought as anti-inflammatory, pro-regenerative. The problem is it's also pro-scarring. So it's sort of, the literature is conflicted because you can't be regenerative and pro-scarring at the same time. And so we're looking sort of a lot of levels below that simplification. We're looking at sort of genetic circuits that could regulate, the antidote to the TGF-beta induced scarring. I should mention that, like in a wound, right, I called TGF-beta the poison. I mean, that's harsh, right? Because TGF-beta is good. Like it does a lot of good stuff to a lot of different cells. It just turns out to be the villain in this story. It's the hero in other stories, right? And so that's why I think the axolotls developed an antidote strategy to keep the good part of TGF-beta and negate the bad. We just don't know what it is that's antagonizing that pathway. Hope that answers that.
We did. Given that axolotls retain their larval traits in adults, do you think their advanced regenerative capabilities are directly tied to their youthful state? And how might we translate this youthful immune environment to adult humans?
Right. So you've opened up a small can of worms, but let me try and explain it. So there's, like, I don't know, 700 or so species of salamanders, ranging from the little tiny guys that fit on my thumbnail from Madagascar. Why is everything so small in Madagascar, right? And then these giant salamanders that are like, like 3 or 4 foot long in places like China and Japan, called giant salamanders, of course. And so there's a lot of different body plans and sizes. But if we look at the limb regeneration field, we're struggling to find one out of, I think, 30 or 40 that have been tested across a range of body plans and types of salamanders that fail to regenerate their limbs as adults. And in fact, all of the newts, which are very closely related to axolotls, they all regenerate their eye tissues and every other body part that's been tested. So they go through metamorphosis naturally. So the idea of post-metamorphic salamanders regenerating is perfectly compatible with all of the salamanders, terrestrial salamanders, and all of the newts. They all regenerate post-metamorphosis as adults with adult immune systems. So that's one kind of line of evidence. I mean, there are problems with metamorphosing axolotls because you're pushing them through puberty very quickly, unnaturally. And so we don't know how that impacts the regeneration circuits. But we can only look into the nature, and it seems like adult regeneration is compatible post-metamorphic salamanders. So I don't know how to get an axolotl all the way through metamorphosis safely and gently and maintain 100% regeneration, but it's something we can consider.
Have you had the occasion ever to look at a crush injury of the spinal cord rather than a clean section?
It's definitely on our list. Yeah, I mean, if anything, it'll probably make it even worse, I think, because once you create a crush injury, you get a big zone of necrosis, and macrophages are responsible for the cleanup. And so it's going to exaggerate the phenotype. There's very little kind of necrosis once you amputate. It's like the cleanest situation. Right. So to see such macrophage dependence in such a clean cut is astounding, right? And so that's what's really exciting. Yeah.
I'm interested in that great picture you showed of macrophages in the liver. That's quite impressive. The question is, where are they coming from? Are they developing in the liver itself as the liver develops, or are they migrating from somewhere else? And if so, where else?
Right. Really important questions. And so people haven't really been able to map hematopoiesis very accurately in the axolotl. And because I've been studying the axolotl immune system for many, many years, I guess it's my job, right? And so, because no one else will do it. And so it's not really high impact, for a lot of, for federal agencies. And so it's always been put down my priority list. But it's becoming really, really important in understanding origin and how that may impact healing. And so we know in one of the papers I published 2 years ago, in adults, all of the regenerative macrophages come from the liver. So it's the lifelong source of regenerative macrophages. And so they're kind of locked into a fetal liver stage. If you look at a mouse's development, they go through various stages over time where it's almost like taking seeds from a friend and putting them in your garden, growing the plant over here, and then waiting for it to seed and then giving it to someone else, and then they put it in their garden. That's kind of how hematopoiesis works over space and time. It starts off in like a blood island, and then a little organ just above the heart, and then that goes to the liver, and then in mice it goes to the bone marrow. And so it's kind of like repotting, if you like. And we have been looking into that process now that we have all of these amazing transgenics and this beautiful microscopy. And actually have a summer student this summer that has been taking embryos at various stages and fixing them and clearing them and putting through like 3D imaging modalities. And he'll present in a couple of weeks, I think. And so with Gabby's help, he's been able to get some beautiful images, and we're looking at how those immune cells associate with developing skeletal structures or developing blood vessels or developing neurons. And so it's kind of interesting, because if they're there at the start when those organs develop, they're probably going to be needed when they repair. And so to answer your question directly, they probably land in the liver somewhere at like stage 40, 43, and then that's where they stay, and there's no bone marrow to go to. The fact that we found a caudal patch of hematopoietic tissue is really interesting. I didn't expect that. Like, I thought zebrafish were weird, but it turns out axolotls are.
Where they're different?
Yeah, I mean, we can do it two ways. We can use antibodies, but we probably use HCR technology, which allows us to take an axolotl-specific probe and look for the RNA in that patch. And we should be able to tell what sort of stem cells are there, what stage of commitment they're in, whether or not they can make T cells, or whether they can make erythrocytes, or we'll be able to map that pretty quickly. And so that discovery will hopefully give us a little bit of insight. And we don't need to see where it disappears, because it could be there like later in life. We don't know yet. It's kind of exciting to think about it. But I mean, I should point out that a mouse is really interesting, because when we published our PNAS paper on the macrophage requirement, the editor who published that, he put 2 people in his lab on that project, and he's a big cardiac lab, and they published a paper showing that macrophages are important in the regeneration of a mouse heart. Now, regeneration of a mouse heart, you might say, what? They regenerate? Well, actually, they only regenerate on day 1. So once the mouse is born, you injure the heart, it regenerates. Day 4, doesn't work, scars. And what happens at day 1? Well, day 1 doesn't have any bone marrow. So it could be the transition to bone marrow that screws everything up. And so that's the theory we're running with. And we'll see, because that just means that we published a paper in mouse showing that the bone marrow derived macrophages sort of turn over the old ones that were there from embryogenesis. So you could theorize that a replacement strategy could be there, like a top up. You could, recharge your cell phone and your blood system at the same time, potentially.
The difference between the macrophages that are developed in the liver and those that are developed in the bone marrow, I mean, they have completely different roles. And do we
Yeah.
continue to produce macrophages from the liver that might have more of a sort of regenerative?
Right. So it's interesting, we do like, so education matters, right? Where you go to school can impact how you think about things. And for macrophages, where they're educated and where they grow up could impact their life choices. I'm going to hunt down pathogens, or I'm going to go, to business school and rebuild organs, right? And so I think it's environment matters. And so what's interesting is there are cases where cancers of the bone marrow shut down bone marrow production of the monocytes. And there's this, like, a few reported examples in medical journals where macrophage production fires back up again in the liver. So there's some latent capacity there, but I mean, realistically, we produce so many monocytes, it might be easier to just modify them and put them back. But, yeah.
Other questions? I have a couple more. I'm just curious, can you explain just a little bit to us about the differences between the two immune systems, the axolotl immune system being so innate and adaptive, and maybe what the trade-off is and why those differences?
Sure. So we've been trying to understand, like, the relationship between the adaptive immune system and regeneration for quite a long time. And so what is the adaptive immune system? Well, it's all of the cells that learn from prior exposure. So when you get vaccinated, these are the cells that remember the previous vaccine, and they produce large numbers of memory cells that are ready to respond at a moment's notice. So the T cells and the B cells, all those sorts of cells are called adaptive immune cells. And so it doesn't seem like axolotls have any real significant population of those cells that could learn, which, puts them at risk of being vulnerable. For instance, if they were under attack from a virus, which is the main line of defense, would be the adaptive immune cells. That helps us track down a cancer cell or a virally infected cell and eliminate that before it becomes a major threat. Salamanders don't have those cells in great numbers. We couldn't find them anywhere. Even single cell sequencing shows that there's, unbiased single cell sequencing shows that there's not many in the animal. And that makes sense because they're quite vulnerable to viruses, right? So that all tracks. But why is that important? Well, those cells are actually sort of important in like recognizing cells that are odd and different from the normal status quo cells, right? And so in frogs, they lose the ability to regenerate right at the moment where they go from the axolotl type of immune system to the human type immune system. They get this massive rise in all of the T cells and B cells that you and I have. And that correlates with the loss of ability to regenerate. But that in itself wasn't really enough, right? That sort of implies that maybe they may have some association with inhibiting regeneration. But in my mouse lab, we actually tested that formally. And so what we did is we took mice and we used mutants that don't produce any T cells, and we found enhanced regeneration. And we've actually have a paper in second review, where we show that it's not just the T cells are bad. It means we found one type of T cell which is absolutely destructive for regeneration, and we found one cell that tempers that behavior and keeps it in check. So it's not a complete loss of regeneration in some contexts, but there are kind of checks and balances in place. We also published a previous paper on this cell type. It's called natural killer cells. And I bet you can guess what they do, right? They hunt down cancer-infected cells and they kill them, right? And so what we found is that there was two versions of that cell, one good one and one bad one. One of them actually helped regeneration, produced all these nice cytokines, proteins, but the other one actually killed the progenitors. So you can't regenerate if the cells that you need to regenerate are getting extinguished. And so that means that like we have cells that are getting in the way of our full potential, right? And so to solve regeneration, we're going to have to basically knock down many of the barriers, right? Fibrosis is one of the barriers. Our overactive adaptive immune system could be another barrier. All of those challenges are solvable with the right, cutting edge therapies. We can transiently attenuate T cells, and we can purify T regulatory cells and then expand them and put them back in, which would tip the balance in favor of regeneration.
I know this is very basic, but after amputation of a limb or section of the cord, what is the signal that causes aggregation of the macrophages?
What is the signal that recruits them?
Molecule that causes aggregation at the site of injury.
Yeah, so recruitment from the blood to
Yes,
the
yes.
and migration. So yeah, the sequence of events is pretty cool actually. So I would say the first thing is, when there's the whole bunch of physical characteristics. So obviously the severing creates like a pulling biophysics signal, probably upstream. There's probably a hydrogen peroxide kind of signal that happens. There's probably an ion current thing that happens. All this is upstream. And then there's a whole bunch of sort of oxidative stress molecules released. And that is the signal for the skin cells, the epithelial cells, to migrate. And they don't proliferate, they just walk really fast, right? And those cells migrate like 10 times faster in axolotl than in human. And then once the seal is made, they start producing these little proteins, like MCP1, that sort of disperse through the blood and through the vasculature. And then all the macrophages that are in the liver or the spleen, they'll smell, what's that? They'll smell it, right? And they'll follow their nose until they get to the strongest source of those proteins. They're called chemokines, and they attract all of the macrophages to the site of injury. And the process is stunning, actually. You can put little beads with those chemokines over here, and they'll go that way instead of this way, and you can direct traffic that way. It's pretty powerful stuff. Yeah.
James, you showed us some pretty impressive imagery in your presentation. So if you sort of travel back in time to your postdoc self, and you think about how technology has changed over that period of time, or what misconceptions or maybe misunderstandings about what you thought then in terms of this process, how is that informed, the evolution of technology, and what are you most excited about for the future in terms of this technology?
Yeah, I mean, I guess microscopy and movies do two things, right? 3D imaging, obviously associated with that. They really overcome that barrier that we all have as scientists about sort of making incorrect assessments of where things are in space. And so back then we were restricted to tissue sections, right? And so, there's two ways to do tissue sections. One's in wax and the other one is frozen. One's, and the thing is, both of them, you can be, done really quickly, or you can be, just everything shreds and your day gets ruined. It can be quite a frustrating process. And so getting serial sections sometimes can be challenging, especially when there's bones involved. The knife hits it and it shatters. And so those kinds of approaches, you don't get every slice. And so you get like fragments, glimpses, snapshots. And then you've got to try and put it in context. Where am I in the tissue? And it's incredibly challenging to make those assessments. Once you get into 3D imaging, you can see the spatial relationship between things. and everything becomes more clear. And in fact, one of the interesting things I remember back in one of my postdocs is we were challenged with this question of whether macrophages and stem cells were actually connecting through these tunneling nanotubes, right? And one of the hypotheses was that there's this cell that was sending out this big javelin over here and then communicating with this other cell over here. And that's what we thought was happening with like the static pictures. When we did live imaging, it turned out to be something completely different. What actually happened when you watch the movie is hilarious. The cell went over to the other cell, put its foot down and went, "How you doing?" Oh, and then walked away, but its foot got stuck. It stretched. So when you look at it as a picture, it looked like a tunneling nanotube, but actually it was just a little foot process that got stuck to the plastic and stretched out. And so that's a funny example of how live imaging can make all the difference. And what you think's happening is not really happening. And seeing is believing, right? So that's, yeah.
James, thank you so much. This is really exciting. We can't wait to hear the rest of the story and figure out the answers to all these questions. That's wonderful. Thank you. So our next Science Cafe, for those of you that are following along, is August 17th. And we are very excited to welcome back a former student, someone who was a student with us about 25 years ago at the College of the Atlantic. And he is now an assistant professor at Duke University. So he's collaborating with one of our scientists here, and they are really looking at kidney disease of unknown origin. So there is this, Nishad Jayasundara is, again, working at Duke to really understand a population in Sri Lanka that is having sort of spontaneous chronic kidney disease. And he's trying to understand where that's coming from. And it's likely a source of environmental pollution and toxins. And so it's sort of just been a really interesting story about utilizing zebrafish as a model for understanding this disease. So we're excited to hear from him, have him back with us, and kind of hear a little bit about his career trajectory since he was here at College of the Atlantic. So I hope you'll all join us and welcome him back. And then for those of you who are around for the next couple of weeks, this is a busy time at MDIBL. So next week we have our annual meeting on the 23rd. You're welcome to join us. We start around 10 in the morning and have a lovely update from Dr. Haller in terms of some of the exciting work that's happening. And we will have a presentation by one of our recent Ph.D. awardees. She received her Ph.D. about 2 weeks ago. She's here with us, has been with us for about 4 years, and is a student at Hanover Medical School in Hanover, Germany, but doing her postdoctoral work, her graduate work here at MDIBL. So she's going to talk about a project that she's working on, which is muscle loss or sarcopenia, and the differences between female and male muscle loss. So it's a very interesting project. She actually looked at the effects of intermittent fasting on preventing muscle loss. And so it's a great project. So I encourage you all to come. We'd love to have you, and you can join us for lunch after. But thank you all so much for being with us this evening. And thank you again to James for coming.